नेक्स्ट रिवाइज कीजिए 9.2.2 प्लांट ब्रीडिंग फॉर डिजीज रेजिस्टेंट। जी हाँ, प्लांट ब्रीडिंग फॉर डिजीज रेजिस्टेंस को रिवाइज कर लिए। चलिए द फर्स्ट क्वेश्चन, स्पेशली कौन से क्लाइमेट में वाइड रेंज ऑफ फंगल, बैक्टीरियल एंड वायरल पैथोजन्स अफेक्ट करते हैं द यील्ड ऑफ कल्टीवेटेड क्रॉप्स स्पीसीज को। Crop losses can often be significant up to -% या कुछ समय इवन टोटल। इस सिचुएशन में डैश डैश एंड डैश डैश ऑफ कल्टीवार्स रेजिस्टेंट टू डिजीज enhances food production. This also helps reduce the dependency on use of dash dash and dash dash. Resistance of the host plant is the ability to prevent the dash dash from causing disease and is determined by the genetic constitutions of the host plant. Before breeding is undertaken, it is important to know about the causative organism and the mode of transmission. Some of diseases caused होते हैं by fungi or rusts. एग्जाम्पल बोलिए इसका। रिवाइज कीजिए मेथड्स ऑफ ब्रीडिंग फॉर डिजीज रेजिस्टेंस। Breeding is carried out by the conventional breeding techniques described earlier or by mutation breeding. The conventional method of breeding for disease resistance is that of dash dash and dash dash. Its steps are essentially identical to those for breeding for any other dash dash characters, such as dash dash. Various sequential steps are screening dash dash for resistance sources, hybridization of selected parents, selection and evaluation of the dash dash and testing and release of new varieties. टेबल 9.1 का माइंड कैप्चर कीजिए। चलिए 9.1 टेबल का माइंड कैप्चर कर लिए। चलिए हम आपको क्रॉप का नाम बताएंगे, आपको उसका वैरायटी बताना पड़ेगा और वो वैरायटी किस डिजीज़ के लिए रेजिस्टेंस है उसको बताना पड़ेगा। चलिए बताइए, वीट क्रॉप वैरायटी बताइए और ये किस डिजीज़ के लिए ये वैरायटी रेजिस्टेंस है, उसको बताइए। नेक्स्ट, ब्रासिका वैरायटी बोलिए। रेजिस्टेंस टू डिजीज। जी हाँ, रेजिस्टेंस टू डिजीज बताइए। कॉलीफ्लावर वैरायटी बदलिए। रेजिस्टेंस टू डिजीज। काउपी वैरायटी। रेजिस्टेंस टू डिजीज। चिल्ली वैरायटी। रेजिस्टेंस टू डिजीज। Conventional breeding is often constrained by the dash of disease resistance gene that are present. Disease resistance gene that are present and identified in various crop varieties or dash. Including dash in plants through diverse means, and then screening the plant materials for resistance sometimes leads to described genes being identified. Plants having these desirable characters can then be either multiplied directly or can be used in a dash. Other breeding methods that are used are dash among dash and genetic engineering. चलिए नेक्स्ट पैराग्राफ रिवाइज कीजिए। Dash is the process by which genetic variations are created through changes in the base sequence within genes. जैसे कि आपने चैप्टर 5 में देखा था। Dash is the process by which genetic variations are created through changes in base sequence within genes, resulting in the creation of new character or trait not found in the dash type. It is possible to induce dash through use of chemicals or radiations like dash radiation and selecting and using the plants that have the desirable characters as a source in breeding. This process is called dash breeding. In a dash resistance to yellow mosaic virus and powdery mildew were induced by dash. several wild relatives जी हाँ several wild relatives of different dash dash species of plants have been shown to have certain resistance जी हाँ certain resistant character but have very low yield. Hence, there is a need to introduce the dash dash genes into the high yielding cultivated varieties. Resistance to yellow mosaic virus in dash dash, that is dash dash, was transferred from a wild species and resulted in a new varieties of dash dash called dash dash. All the above examples involve source of dash dash that are in the same crop species which has to be bred for disease resistance or in a related wild species. Resistance gene का transfer is achieved by dash dash between the target and the source plant followed by selection.